this particular intellectual property that has been developed by my group and I, along with collaborator, Professor Xuanhe Zhao, who is now a professor at MIT. And the general patent is called something like Devices and Methods for Active Biofouling Control. Biofouling here basically refers to the accumulation of microbial communities on surfaces that are immersed in aqueous solutions of any sort, where you have viable microbes and some sort of nutrient source. So, as noted on the slide, they are ubiquitous and can be found across nature, natural and industrial and medical types of scenarios. Here you see, for example, a biofilm in a section of pipe. You can also see a natural biofilm that occurs in streams in Yellowstone National Park, for example, on the surface of teeth, in industrial seawater cooling plants that, for example, here on the bottom middle, and then on the surface of ships. What I've been asked to focus on today is the problem of biofouling of urinary catheters. So that's shown here on the bottom left. So basically, the formation of a biofilm or biofouling by microbes is really an integral part of their life cycle of the microbial community. You can think of these biofilms as sort of cities on the surface of materials that the microbial communities that inhabit them will exist. They like to form these films to protect themselves from predation, to allow efficient genetic exchange, and also, as it turns out, increase their or decrease their susceptibility to antibiotics. And so, from a health perspective, they're quite problematic, because once they form, and oftentimes they do form on implanted materials, and so they can be quite dangerous and tough to deal with. There's been a lot of ways in which people have tried to deal with them on implant materials, for example, by developing materials that exude antibiotics or are just hard for bacteria or other microbes to colonize, but in general, it's a pretty tough problem that has not really been solved. Our innovation was to note that if you have a biofilm on an elastomer substrate and you stretch that elastomer or deform it somehow, you can effectively debond the biofilm. So that's what we'll be talking about today. And so it's sort of an analogy to the destruction of a city. So as I mentioned, I'll be focusing on this problem of urinary tract infections on catheters, so-called Foley catheters. This is a major problem in hospitals and other care facilities where you have to have long-term catheter insertion in a patient. So the picture here shows what a Foley catheter typically looks like. It's basically a rubber device that has a couple of lumen in its cross-section, as shown in the bottom right. Basically, one of those lumen, the larger one, is where the urine flows. And then the other smaller lumen is what's called an inflation lumen. And that's used to inflate this balloon that, once it's inserted into the bladder, is inflated to hold the catheter in place. So given that we discovered this phenomenon by which deformation of a surface can lead to dislodging of the biofilm, we had this idea to develop a Foley catheter that would have more than just the 2 lumens, but would have additionally some what we call inflation lumens at the periphery of the urine drainage lumen, so that after some time, when you get biofilm formation on the urinary drainage lumen, you can actuate the inflation lumens to deform the surface of the urine drainage lumen and thus disrupt the biofilm and flush it out. And the nice thing about this approach is that these catheters are already made of elastomers like silicones or urethanes or natural type of elastomers. So that we don't really need to develop new materials. The material set is there already. So for our proof of concept studies, we developed this, or we used this artificial bladder and catheter prototype that's indicated here. Basically, the supply reservoir here would be inoculated with whatever sort of infectious microbes we are interested in studying. And the first study we published on mixed culture of Proteus mirabilis and E. coli, which are known to form problematic biofilms on urinary catheters. In the subsequent study, we used yeast, Candida albicans, as the model organism. And so basically, you put them into the supply reservoir and you slowly let them drip into this artificial bladder, and then this artificial urine that's infected drips down the catheter, and that is actually sort of like the perfect storm for formation of these biofilms inside of these catheter prototypes. And so here are the results, basically for on the left for the bacteria, on the right for the yeast. And basically what you can see is that we've, after we're done with the experiment, we stain the bacteria or the yeast with a purple dye. And you can see that in the control, you have a good biofilm, a good amount of biofilm formation, biofouling of this lumen. But if you inflate it a few times, you can dislodge that biofouling very effectively at flow rates that would be commensurate sort of with the drinking of a large glass of water. So you see that for both the bacteria and for the yeast biofilm, which actually have very different structures that I'm not showing here. So that's basically the idea, is that we would be able to inflate these inflation lumens, deform the lumen that's carrying the urine, and dislodge the biofilm from that lumen. And in principle, we can do this over and over again. And so what you see here on this slide is a video of the inflation in process for a prototype. And also you see a fairly well-developed design for our multi-lumen Foley catheter here that would be designed for combating these infections. So again, the one port the top port would go to inflate the balloon. The middle port would be connected to a urine drainage bag, and then the bottom port would be the port that would be used to inflate the inflation lumen. And so the applications are basically to control catheter-associated urinary tract infections without any need for any harsh treatments. or any antibiotics. Of course, this could be used in concert with antibiotics. The materials are nearly identical to those of current catheters, so that is quite useful. And I should point out that through extrusion molding, it's fairly easy to manufacture these catheters, and as you can see in our prototypes. There could be potentially other uses beyond Foley urinary catheters, including other types of medical catheters, but also, as I suggested in the beginning, in other medical, industrial, or military applications. As far as catheters, there's 30 million Foley catheters used per year in the US. 10 million of those are used for long-term catheterization, which increases the risk of the formation of these biofilms and therefore infection. This slide also gives data on central venous catheters and PIC catheters as well. So the current status is that we have in vitro testing that clearly shows the efficient removal of diverse biofilms. Next steps are indicated here as well.